Hello bachcho, welcome to Hello Biology. This is the third lecture of the chapter Principles of Inheritance and Variations. In the previous lectures, we understood Mendel's monohybrid experiment. Now it's time to move one step deeper into genetics and understand how Mendel verified his results and explained the inheritance of multiple characters together. Today's lecture is going to be very interesting because we will explore some important genetic experiments and concepts that form the strong foundation of inheritance. So get ready to think like a geneticist and understand biology in the simplest way possible. Now, students, let us start with another very important concept in genetics, test cross. Mendel had already understood that dominant traits appear in the offspring very easily. But then one important question came into his mind. Suppose we see a tall plant. Can we say with complete certainty that this plant has only dominant factors? Maybe the plant is pure tall, or maybe it only looks tall because dominant trait is hiding the recessive one. So how can we find the real genetic makeup of that organism? To solve this problem, Mendel performed a special type of cross called test cross. A test cross is used to determine whether an individual showing a dominant trait is homozygous dominant or heterozygous dominant. In this cross, the organism showing the dominant trait is crossed with a recessive parent. Why only recessive parent? Because recessive traits remain hidden in normal conditions. So crossing with a recessive parent helps reveal the hidden factors present in the dominant individual. This way, Mendel was able to identify whether the dominant-looking organism was pure or hybrid. So remember, students, test cross is mainly performed to identify the genotype of an individual showing dominant phenotype. Now, students, let us understand how a test cross helps us identify whether an unknown dominant individual is homozygous or heterozygous. Look carefully at this example. We have a yellow seed plant. Since yellow color is dominant, the plant appears yellow. But there's a problem. Just by looking at the plant, we cannot tell whether it is homozygous dominant, YY, or heterozygous, Y small y. So how can we find out its real genotype? To solve this problem, we perform a test cross. In a test cross, the unknown dominant individual is crossed with a homozygous recessive parent. Here, the unknown yellow seed plant is crossed with a green seed plant, which is homozygous recessive Y small y. Now observe the result. After the cross, all the offspring produced are yellow. Not a single green offspring appears. What does this tell us? If the unknown parent had contained a recessive factor, some green offspring would have appeared. But since every offspring is yellow, it means the unknown parent can give only dominant factors. Therefore, the unknown yellow plant must be homozygous dominant, Y capital Y. So, students, remember this important point. If all offspring show the dominant trait after a test cross, the unknown parent is homozygous dominant. In this example, yellow times green, Y small y, equals all yellow offspring. Therefore, the test plant is homozygous dominant, Y capital Y. This is how Mendel used a test cross to reveal the true genetic makeup of an organism. Now, students, let us look at the second possible outcome of a test cross. In the previous example, all offspring were yellow, and we concluded that the unknown plant was homozygous dominant. But what if the results are different? Let us see. Again, we start with a yellow seed plant whose genotype is unknown. We know that yellow color is dominant, but we do not know whether this plant is homozygous or heterozygous. To find out, we perform a test cross. The unknown yellow plant is crossed with a homozygous recessive green plant, YY. Now observe the offspring carefully. This time, the F1 generation contains both yellow and green offspring. Out of 4 offspring, 2 are yellow and 2 are green. This gives a ratio of 1 yellow to 1 green. Now think about it. If the unknown plant were homozygous dominant, all offspring should have been yellow, but green offspring are also appearing. This means the unknown plant must be carrying a recessive factor along with the dominant factor. Therefore, the unknown yellow plant is not homozygous dominant. It is heterozygous. YY. Check. So, students, remember this important rule of test cross. If all offspring show the dominant trait, the test plant is homozygous dominant. If the offspring show a 1-to-1 ratio of dominant and recessive traits, the test plant is heterozygous. In this example, yellow times green, YY, equals 1 yellow to 1 green. Therefore, the unknown test plant is heterozygous, capital Y and small y. Check. This is how a test cross helps us reveal the hidden recessive factor present inside a dominant-looking individual. Let us discuss the results of the test cross and understand what Mendel observed from this experiment. Mendel performed test crosses to discover the true genetic makeup of individuals showing a dominant trait. At first glance, two dominant-looking plants may appear exactly the same. For example, two yellow-seeded plants both look yellow from the outside. But Mendel realized that appearance alone cannot reveal the actual genotype of an organism. So he crossed the dominant individual with a homozygous recessive parent and carefully observed the offspring. After performing many test crosses, Mendel made two important observations. First observation. Sometimes all the offspring showed the dominant trait. For example, when a yellow-seeded plant was crossed with a green-seeded plant, all offspring were yellow. From this result, Mendel concluded that the unknown parent was homozygous dominant. This is because a homozygous dominant individual can pass only dominant factors to its offspring. Therefore, no recessive phenotype appears in the next generation. Second observation. In some cases, the offspring appeared in a ratio of 1 dominant to 1 recessive. For example, half of the offspring were yellow and the other half were green. This result indicated that the unknown parent was heterozygous. Mendel concluded that the dominant-looking parent contained both dominant and recessive factors. During gamete formation, these factors separated and passed into different gametes. As a result, some offspring received the dominant factor and appeared dominant, while others received the recessive factor and showed the recessive trait. What did Mendel learn? From the test cross, Mendel learned that 1. The appearance of an organism does not always reveal its genotype. 2. A dominant phenotype can be either homozygous dominant or heterozygous. 3. Recessive factors can remain hidden in an individual and can be detected through a test cross. 4. A test cross is a reliable method to determine the genotype of an individual showing a dominant phenotype. So students, the most important conclusion of test cross is this. Test cross helps us uncover the hidden genetic makeup of a dominant-looking organism from a dominant-looking organism. That is why test cross became one of the most important tools in classical genetics. Genetics.
Students, till now we have studied how a single character is inherited from parents to offspring through a monohybrid cross. But Mendel was not satisfied with understanding only one character at a time. He started wondering, what happens when two different characters are inherited together? Do they always travel together from parents to offspring? Or does each character behave independently? To find the answers to these questions, Mendel designed another fascinating experiment known as the Dihybrid Cross. And believe me, students, this was not just another experiment. This experiment opened the doors to some of the most important and hidden truths of genetics. Through this experiment, Mendel discovered how multiple traits are inherited simultaneously and how they behave during the formation of offspring. The observations from this experiment were so remarkable that they helped scientists understand patterns of inheritance that were completely unknown at that time. In fact, the results of the dihybrid cross became one of the strongest foundations of modern genetics. So get ready, students. We are now going to explore one of Mendel's most powerful experiments, an experiment that revealed hidden genetic secrets and changed the way we understand heredity forever.
Let us now explore the dihybrid cross first. First of all, let us understand what a dihybrid cross actually is. Till now, we have studied monohybrid cross, where Mendel observed the inheritance of only one character at a time. But Mendel wanted to know something more. He wondered, What will happen if I study two characters together instead of one? Will both characters be inherited together? Will one character affect the inheritance of the other? Or will they behave independently? To answer these questions, Mendel designed a new experiment called the Dihybrid Cross. A dihybrid cross is a breeding experiment that deals with two heritable characters at the same time. In this experiment, Mendel selected two different characters of the pea plant simultaneously. He chose seed color, yellow or green, seed shape, round or wrinkled. Now, instead of studying only seed color or only seed shape, Mendel studied both characters together in the same experiment. This made the experiment much more interesting because he could observe how two different traits were inherited from parents to offspring at the same time. And students, the results of this experiment were truly remarkable. The dihybrid cross helped Mendel uncover one of the most important principles of genetics and showed that different pairs of traits can be inherited independently of each other. So remember, monohybrid cross, one character studied at a time. Dihybrid cross, two characters studied at the same time. Now, let us move ahead and see how Mendel actually performed this fascinating experiment.
Let us see how Mendel actually performed the dihybrid cross. In this experiment, Mendel selected two pea plants that differed in two characters simultaneously. The first pea plant had round and yellow seeds. The second pea plant had wrinkled and green seeds. Mendel crossed these two pure breeding pea plants and carefully observed the offspring produced. Now, what do you think happened? Did some offspring have round seeds and some wrinkled? Did some have yellow seeds and some green? Interestingly, none of these variations appeared in the F1 generation. All the plants of the F1 generation produced round and yellow seeds only. This observation was very important. It showed that for seed shape, round trait was dominant over wrinkled trait. Similarly, for seed color, yellow trait was dominant over green trait. So both dominant traits appeared together in the F1 generation, while the recessive traits remained hidden. But Mendel was not satisfied with observing only the F1 generation. He allowed the F1 plants to undergo self-pollination and produced the F2 generation. Now, something very surprising happened. In the F2 generation, Mendel observed not one or two, but 4 different kinds of plants. Some plants had round yellow seeds, some had round green seeds, some had wrinkled yellow seeds, and some had wrinkled green seeds. Students, pay special attention here. The combinations round green and wrinkled yellow were not present in the original parent plants. These were completely new combinations that appeared in the F2 generation. This observation made Mendel realize that different pairs of traits can assort independently during inheritance. And this remarkable discovery eventually led to one of the most important laws of genetics, the law of independent assortment. Now, let us see these F2 offspring and understand how these 4 different combinations appeared.
Now let us carefully understand the diagram shown on the screen. At the top, we have the parent generation, also called the pea generation. Mendel selected 2 pure breeding pea plants that differed in 2 characters. One plant had round and yellow seeds, while the other plant had wrinkled and green seeds. These plants were crossed with each other. Now look at the F1 generation. Surprisingly, all the offspring produced round and yellow seeds. No wrinkled seeds appeared, no green seeds appeared. This clearly indicated that round seed shape is dominant over wrinkled seed shape, and yellow seed color is dominant over green seed color. So in the F1 generation, only the dominant traits were visible. Next, Mendel allowed these F1 plants to undergo self-pollination. The seeds produced after selfing formed the F2 generation. Now observe the F2 generation carefully. This time, 4 different types of seeds appeared. The largest group consisted of round yellow seeds. The second group had round green seeds. The third group had wrinkled yellow seeds, and the smallest group had wrinkled green seeds. Students, this is the most important observation of the dihybrid cross. Notice that round green and wrinkled yellow combinations were not present in the parent generation. These are new combinations that appeared in the F2 generation. This showed that seed shape and seed color were behaving independently during inheritance. When Mendel counted all the F2 plants, he found a characteristic ratio: 9 round yellow to 3 round green to 3 wrinkled yellow to 1 wrinkled green. This ratio is called the dihybrid ratio and is represented as 9 to 3 to 3 to 1. From this experiment, Mendel concluded that different pairs of factors separate and combine independently during inheritance. This conclusion later became known as Mendel's law of independent assortment. And students, this is why the dihybrid cross is considered one of the most important experiments in the history of genetics.
Now let us analyze the results of Mendel's dihybrid cross in detail. Look carefully at the F2 generation shown on the slide. Mendel observed 4 different types of seeds. The first and most common group was round yellow seeds. The second group was round green seeds. The third group was wrinkled yellow seeds, and the fourth group was wrinkled green seeds. When Mendel counted all these seeds, he found a very specific pattern. For every 16 offspring produced, 9 were round yellow, 3 were round green, 3 were wrinkled yellow, and only 1 was wrinkled green. This gives the famous phenotypic ratio, 9 to 3 to 3 to 1. Students, this ratio is called the dihybrid ratio and is one of the most important results in genetics. Now, pay special attention to something very interesting. Round yellow and wrinkled green were the same combinations that were present in the parent generation, but round green and wrinkled yellow were completely new combinations. These new combinations did not exist in the parental generation. Such new combinations are called recombinants. In simple words, recombinants are offspring that show a new combination of traits different from their parents. Here, round green seeds are recombinants. Wrinkled yellow seeds are also recombinants. This observation was extremely important because it showed that seed shape and seed color were not inherited as a single unit. Instead, the factors controlling these traits separated and combined independently. Now, let us look at the genotypic ratio shown below. The genotypes are distributed in a definite pattern that finally produces the phenotypic ratio of 9 to 3 to 1. Although the genotypic combinations are many, they collectively give rise to only 4 visible phenotypes. From these observations, Mendel concluded that different pairs of factors assort independently during gamete formation. This conclusion later became known as the law of independent assortment. So students, always remember, 9 to 3 to 3 to 1 is the characteristic phenotypic ratio of a dihybrid cross. Round green and wrinkled yellow are recombinant phenotypes. The appearance of recombinants provided strong evidence for independent assortment, and this became one of Mendel's greatest contributions to the field of genetics. Students, till now, we have discussed the most famous dihybrid cross performed by Mendel using seed shape and seed color. But an important question arises. Did Mendel perform only one dihybrid cross experiment? The answer is no. Mendel repeated the same type of experiment using several other combinations of characters to verify whether the pattern of inheritance remained the same. This was very important because a scientific conclusion cannot be based on just one experiment. So Mendel selected different pairs of contrasting characters and performed dihybrid crosses again and again. Let us look at some of these combinations. The first combination was seed shape and seed coat color. He crossed plants having round gray-brown seeds with plants having wrinkled white seeds. The second combination was seed shape and flower position. In this experiment, Mendel crossed plants showing round seeds with axial flowers and plants showing wrinkled seeds with terminal flowers. The third combination was seed color and flower position. Here, he crossed yellow-seeded plants having axial flowers with green-seeded plants having terminal flowers. And finally, he studied seed color and stem height together. In this experiment, yellow tall plants were crossed with green dwarf plants. Now, students, you do not need to memorize these combinations right now. The most important thing to understand is why Mendel performed these additional crosses. He wanted to check whether the behavior of inheritance was the same for all pairs of characters. And interestingly, the results were consistent. Again and again, Mendel observed that different pairs of traits behaved independently during inheritance.
This strengthened his conclusion and provided strong evidence for the law of independent assortment. So remember, students, Mendel did not rely on a single experiment. He repeated dihybrid crosses using many different character combinations, and all of them supported the same fundamental principle of genetics. That is why his conclusions became one of the strongest foundations of modern genetics. Raise G.
Mendel's dihybrid cross revealed how two different characters are inherited together from parents to offspring. Through this experiment, Mendel discovered that different pairs of traits assort independently during inheritance. The results of this experiment led to the formulation of Mendel's law of independent assortment, one of the fundamental principles of genetics. Now let us focus on one of the most important observations of Mendel's dihybrid cross. When Mendel studied the F2 generation, he found that the offspring were not all identical. Instead, 4 different types of plants appeared. This was a very surprising observation because the F1 generation showed only one combination of traits. Now, let us look at these 4 categories carefully. The largest group consisted of plants showing both dominant traits. These plants had round and yellow seeds. Out of every 16 plants, 9 belonged to this category. The second group showed one dominant trait and one recessive trait. These plants had round and green seeds. Out of every 16 plants, 3 belonged to this category. The third group also showed one dominant trait and one recessive trait, but in a different combination. These plants had wrinkled and yellow seeds. Again, 3 out of every 16 plants belonged to this category. Finally, the fourth group showed both recessive traits together. These plants had wrinkled and green seeds. Only 1 out of every 16 plants belonged to this category. So students, the 4 types of plants appeared in the ratio 9 to 3 to 3 to 1. This is called the dihybrid phenotypic ratio. Now, think about it. If traits always remained together, we would have seen only the parental combinations. But Mendel also observed new combinations, such as round green and wrinkled yellow. These new combinations proved that different pairs of traits can assort independently during inheritance, and this observation became the basis of Mendel's law of independent assortment. So remember, 9 plants, both dominant traits. 3 plants, first dominant, second recessive. 3 plants, first recessive, second dominant. 1 plant, both recessive traits. Together, they form the famous dihybrid ratio of 9 to 3 to 3 to 1. बच्चों, an important observation of Mendel's dihybrid cross was the appearance of 2 completely new combinations of traits, round green and wrinkled yellow. These combinations were not present in the parent plants and are called recombinants. Their appearance showed that different traits can assort independently and recombine in new ways in the offspring. When Mendel performed the dihybrid cross, he observed that the factors controlling seed shape and seed color did not remain linked together. Instead, they separated and assorted independently during gamete formation. As a result, new combinations, such as round green and wrinkled yellow, appeared in the F2 generation. This observation led Mendel to propose the law of independent assortment. That's all for today's lecture, students. Today, we explored some of Mendel's most important experiments and discoveries, including test cross, dihybrid cross, and results of dihybrid cross. I hope this lecture helped you understand these concepts in a simple and interesting way. Thank you for being a part of Hello Biology. Keep learning, keep growing, and keep exploring the fascinating world of biology. Until our next lecture, take care and happy studying. Don't forget to like, comment, share, subscribe. Thank you and goodbye.